Hi, welcome back to the carbohydrate playlist in biochemistry. My name is Kevin Tokoph. Make sure to like this video and subscribe to the channel for future videos and notifications. Alright, so in the last few videos we've been talking about monomers of sugars, which are called monosaccharides, and how we can combine those to make disaccharides, trisaccharides, oligosaccharides, and we're ultimately going to be talking about how you make things called polysaccharides, which are the largest. So when you put 100 to 1,000, somewhere in that range, of monosaccharides together, you get what's known as a polysaccharide. And we're going to do several videos on these, the first of which is this video, which is the topic is going to be cellulose. All right, so where do you find cellulose? Well, cellulose is, first of all, the most abundant source of carbon on the planet. The main place that you find it are the cell walls of plants. So you think of the cell walls of leaves and branches and stems and so forth, and one of the reasons they're so strong is actually not protein. Usually we think of protein making things strong, right? It's actually a carbohydrate polymer in this case that makes the plant strong, and that's actually this, which is cellulose. All right, so the main place you're going to find it is the cell wall of plants. All right. So the first thing to notice about cellulose is what the repeating unit is. All right, some of these polymeric carbohydrates are going to have different monomers. In other words, they're going to have more than one repeating unit. For example, we're going to look at one called peptidoglycan, and the sugar component of that is actually two. It's N-acetylmuramic acid and N-acetylglucosamine. But others like cellulose and chitin only have one, and the question is, what is it? The answer is it's just simple D-glucose in the beta conformation. All right? And you put a bunch of these glucoses together in this linkage fashion, and we'll talk about that in a minute, and that's going to give you the polymer cellulose. So all it is glucose. In the next video, we're going to look at chitin, something that's found in the exoskeleton of insects. That one is not glucose, it's repeating units of N-acetylglucosamine. So you can vary the sugar and vary the combinations of them to get different polymers. And also you can vary the linkages as well. All right? So what's important about the linkages in cellulose? And we'll talk about the linkages and then what the implications of that are. Let's look first at this glucose monomer right here. This is just simple beta-D-glucose. And in fact, I've actually drawn it in the exact same orientation that it will be for every one of these monomers in the polymer cellulose. So let's talk about it. First, I want to look at this carbon right here, the carbon that is essentially, or the group I should say, that is counterclockwise from this oxygen that's part of the ring. And then I also want to look at this group right here. All right. So if I look specifically at this hydroxymethyl group up here, I notice that with respect to the ring, it's going up, right? And if I look at this one, which happens to be the anomeric carbon, I notice it's also going up. And if you think back to some of the previous videos where we talked about alpha and beta, and I recommend if you don't know what alpha and beta are to go back, but when you look and see both of these groups, counterclockwise from the oxygen and clockwise, both going up, we say that this conformation happens to be beta, okay? In other words, we're talking about the anomeric carbon happens to be in the beta configuration. And in fact, to polymerize these glucose molecules into cellulose, this has to be in the beta configuration. It cannot be alpha, okay? It turns out that there are other cases, and I'll go ahead and draw it down here. There's other cases where, in fact, the glucose, when you combine it with another sugar, you actually want it to be in the alpha configuration. So let's talk about that and maybe some implications of that. All right, so let's look at this. So we look at this group that's going counterclockwise from that's oxygen that's part of the ring. We notice it's going up, right? We can also look at this group right here. This also happens to be the anomeric carbon because this is also glucose, right? This is D-glucose. Is it beta or alpha though? Well, this group is going down. And when this group whenever this group on the anomeric carbon or whatever is clockwise from that oxygen is going the opposite direction as what's going counterclockwise, up and down, this is instead alpha. So this carbon we call alpha, the anomeric carbon. So this is alpha D-glucose. It turns out to make some sugars like maltose, which is a dimer of two glucose molecules, this carbon right here ought to be in the alpha configuration, and it actually has to be. Whereas when you make cellulose, this carbon has to be in the beta configuration. So this one has to be beta. And in fact, if you look at all of those carbons on this chain, they're all beta. Every one of them in cellulose is. It's impossible for one to be in the alpha configuration. Okay, so hopefully that makes a little bit of sense. Now, the question is, what are some implications of this being in the beta configuration? All right, there's two things, and we're also going to talk about these same two things with chitin in the next video. Chitin, it turns out, is very similar to cellulose in this respect, and so it may seem repetitive in the next video, but it really is the same. All right, so what are the implications of that beta configuration of this carbon right here? Okay. Number one, and I'm actually going to write these up here. The first implication has to do when you look at these all these sugars in cellulose. When you look at them in the chair conformation, so let me first write that. We're not going to do that in this video, so for now you're going to have to take my word for this, but I will show you in another video what this looks like, because it's really complicated and I don't want to convolute the issue here. But it turns out when you look at all of these in chair conformations, it turns out that because of the beta configuration of this anomeric carbon for every one of them, there is extensive hydrogen bonding. Okay? Now, what does that extensive hydrogen bonding do to the stability? It drastically increases the stability of that carbohydrate. In this case, it's cellulose. It turns out that for chitin, it's going to do the same thing, except we said the monomers were N-acetylglucosamine, not glucose. In cellulose, they're glucose. So if you were to look at this in the chair conformation, and you can actually, if you wanted to go ahead and just do that, you could pause the video and probably find it on a quick Google search. And what you'd see is that some of the pictures will actually depict the hydrogen bonding, and there's a lot of it. And hydrogen bonding adds to increased stability of the polymer, which makes it very, very strong. And we already said that cellulose is what gives plants a lot of their rigid structure. It's the main constituent of their cell walls, and it makes them very strong. The strength has a lot to do with their hydrogen bonding as well. Okay? Now, if we're looking at this in terms of a physiological perspective, so let's say you were to eat this, okay? In fact, you do. Whenever you eat something that has vegetables, okay, even if you eat a burger, right? If you eat a burger, you know, if you have some vegetables on it, like onions, lettuce, there's less cellulose in that than there is in, you know, a tree branch, but there is cellulose. So the question is, what's happening to it, all right? Nothing happens to it, all right? So, when I say this, I'm specifically talking about in humans, okay, because this is generally what we're concerned with. In humans, there are no enzymes in general to digest, or we could say break, beta carbohydrate linkages. So what does that mean? Well, remember, whenever you eat something, okay, it goes into the stomach, there's the acid there, and there's some enzymes like pepsin there. Okay, that's beside the point. Then those things pass into the small intestine, and that's where the main enzymatic degradation occurs. However, in humans and many other animals, which mainly include carnivores, we don't have enzymes that degrade polymeric carbohydrates with beta linkages, okay? So the fact that there's a beta linkage right there in cellulose, there's beta, all these are beta. The fact that it's beta means that this polymer is completely indigestible by enzymes in humans, okay? When we look at chitin, because there are cultures that eat insects, and if you go into the jungle, we can't survive eating the plants there, okay? So you would default to eating insects, so you could do that. The chitin in insects also has this beta linkage and therefore is also indigestible by humans. So the question is, can I derive energy from glucose from cellulose or chitin? The answer is no. You can't break these beta linkages, so this polymer does not get digested. It doesn't get broken down, so you don't derive energy from it. A common term you may have heard for this kind of substance is fiber. Okay, that's a pretty common term that we hear in dietary language, and in fact cellulose is a fiber. In general, what fiber means from this perspective is it's some kind of polymer that is completely indigestible by humans. Meaning it goes into the stomach when we eat it, then it goes into the small intestines, it can't get degraded, so then it keeps passing through. It'll go to the large intestine and ultimately out into the toilet. It doesn't do anything. It can't get absorbed because it's far too large. Okay? It can't get broken down either. And that's because it has this beta linkage, which, at least from a physiological perspective in the digestion, it gives it increased stability that way as well. However, the difference is when we have something like maltose, which has an alpha linkage. I want to make that perfectly clear. This is alpha. Okay, remember this group is going up, this group is going down, therefore it's alpha. All right. Turns out maltose is only a disaccharide, but it has the same linkage as starch, which is a polymer. Okay, starch has many glucoses bonded together. Starch is what you find in like potatoes, and starch has every one of those in an alpha linkage too. We're going to have a separate video on starches also. But the point is, humans do have enzymes to break alpha linkages. Okay, if this is the carbohydrate or disaccharide maltose, we have an enzyme called maltase to break it down. And since this is two glucoses bonded together, when we break it, we just get two glucoses back. So this is glucose, okay? We get two of those back, all right? So what's the point? Anytime we have carbohydrate polymers, and it could be something as simple as a dimer like this. The point is when there's multiple carbs bonded together and they're in an alpha configuration, we have the potential to degrade that and get energy out of it. But whenever it's in the beta configuration, like this, we can't break those down and therefore we can't get energy from it. So those are termed dietary fibers. Okay, and chitin is also going to be an example of that in the next video. Now, one other thing I wanted to do very quickly is I wanted to discuss the nomenclature of this, even though we already discussed how to talk about this in a previous video. I just want to discuss it. And to do that, what I'm going to do is I'm going to look at these carbohydrates and I'm going to number each carbon. Okay, and this is actually the this is going to be the absolutely correct way to number these. You can number them for some things any way you want, but when you do this linkage stuff, you have to number them a certain way. Okay, and this is the way I'm numbering glucose is the way that you number it. All right, when you number the glucose, this oxygen in the ring does not actually get a number. Okay, in general, because it doesn't link to other carbohydrates. All right, so beta 1 to 4. So what I'm going to do is I'm going to generically just pick one of these. I'm going to pick this carbohydrate and look at that. So I look at this carbohydrate and I look at its anomeric carbon, which happens to be in the beta configuration. And that's important because it is the anomeric carbon that's bonding to other things. All right, so I look, this is going up, seems to be going up, so it's beta, right? So I write a beta here, and whichever atom has the beta. So I look and it's the anomeric carbon, which happens to be according to the absolute numbering system. This is carbon 1, so it's beta 1. In other words, carbon 1 is in the beta configuration. That's why it's called that. Then you draw this arrow to the right and to a number, okay? The number that you draw it to is the number on the next carbohydrate of that carbon that it attaches to. So 1, 2, 3, 4 on the next unit. It attaches to position 4. So a carbon 1 in the beta configuration connects to a carbon 4 in the next carbohydrate, okay? And what you would find is that every one of these linkages for cellulose is beta 1 to 4. Also for chitin, it's also beta 1 to 4. There's only one difference in that chitin has N-acetylglucosamine as the subunit, whereas cellulose has glucose as each individual subunit, okay, or monomer. All right, and that's about it that we have to talk about cellulose. Join us in the next video where we talk about chitin, and then after that we're going to talk about some other kinds of polymers such as amylose and amylopectin, and then we'll conclude with glycogen. All right, so thanks for watching this video. I hope it helped, gave you some insight. Make sure to like the video, subscribe to the channel for future videos and notifications. Thank you very much.